Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today. Today we have the honor of being joined by Dr. Eddie Holmes, who is an evolutionary biologist who's distinguished for his work on both the emergence and evolution of viruses. And over the years of his career, he has used genomic and phylogenetic approaches to reveal major mechanisms of virus evolution and has determined the genetic and epidemiological processes that explain how many viruses jump species boundaries, spread to new hosts. Dr. Holmes' work has revealed the origin, evolution, and molecular epidemiology of very important human pathogens, including influenza, HIV, dengue, and has enabled accurate assessments of what types of viruses are most likely to emerge in human populations, whether they're evolved for human-to-human spread. And his recent research has provided really fundamental insights into a breadth and biodiversity of the viral world, and has additionally studied the emergence and spread of pathogens including COVID, Hepatitis C, Yersinia pestis, as well as myxoma and rabbit hemorrhagic disease virus, among many others. Dr. Holmes is currently a professor in the Sydney Institute for Infectious Diseases, School of Life and Environmental Sciences, and City Medical School at University of Sydney, and his previous appointments have included as the Vern Wayman Chair in Life Sciences down the road here at Penn State University, as an affiliate member of the Fogarty International Center down the road at the NIH. He was also a fellow from 1999-2004 at New College, Oxford, honorary visiting professor at Fudan University in Shanghai, and he is the author of close to 700 peer-reviewed papers, including 2 books. We're honored to have him with us today. Dr. Eddie Holmes, thank you so much for taking the time to come on the show.
Oh, my pleasure.
It's great having you here. As I mentioned in the intro, across the hundreds of papers that you have published in the peer-reviewed literature over the years, while you've been focusing on these core themes of viral evolution and emergence, unlike many of our previous guests who specialize in a few of the usual suspect viruses mentioned in your bio, your portfolio of viruses is quite extensive. In addition, to what I mentioned, you've published on rubella-like viruses in the Pacific Electric Ray, in picornaviruses in New Zealand reptiles, and one of my favorites, the insect-specific alpha virus, the yada yada virus, just to name a few.
Yeah.
Talk a little bit about when the Virosphere, and this is a theme we'll get back to quite a bit, is your target. How do you decide ultimately which of these bugs you're working on in any given day?
Yeah, that's a very good question. So as you said at the start, I think I'm more of an evolutionary biologist than a virologist, right? So the questions that have always driven me are evolutionary questions. So I want to understand the diversity of nature and what explains that diversity. And so when I think about understanding viruses, my focus is always trying to what's the evolutionary angle. And so the species that we tend to sample tend to be ones that can address interesting evolutionary questions, okay, or ecological questions. So, for example, we've just been to New Zealand in the Fiordland. It's a beautiful place, right in the far kind of southwest corner of New Zealand, very remote. It's Lord of the Rings kind of territory. It's amazing remote islands. We've been to one little island there called Anchor Island, which is you can only get by helicopter. And we basically sampled every species on the island. Okay, so every bird, every reptile. Luckily, it's only one reptile. every mammal. There are no mammals. Okay, lots of insects. And the idea it's a whole ecosystem. And the idea is, how do viruses flow between an ecosystem? And so that meant sampling things like kiwi are there. Obviously, you know what kiwis are. Kakapo, they're these really rare parrots. A thing called a moorbuck, which is a moorpork, sorry, which is like a New Zealand owl. So and so these were species that you never think about, but because they were ecologically linked, we decided to look at them. So again, the species that I sampled were entirely based on what's the evolutionary ecological question.
And as you sample some of these sort of lesser sampled species, and obviously, you know, as mentioned, you know, core to your work is, you know, understanding this host species barrier issue with viruses, you know, what's responsible for transmission, what's responsible for protection. And obviously, you know, if we think of the usual suspects again, whether it's that chicken or the monkey or the rat, whatever it may be that we think about in terms of some of these pathogens that impact us as humans, you know, you also publish on the platypus, the penguin, the bottlenose dolphin, and here we got birds and mammals and other types of things that are quite similar. What are some of the top line learnings in the sense, you know, why the chicken, why not the penguin, and some of the top line insights that you
why
see?
Why did the chicken get a virus?
Exactly.
So look, that's a good question, but I think you need to understand. So the way I see it is that so what we've gone through with COVID, right? You know, is because a virus jumped the species boundary, and that process happens. It you know, jumped from whatever it was, may have bounced with an animal into a human and caused a new disease, and that happens on a regular basis. And so we, as humans, our focus on that is on the kind of very much the health and economic consequences of that process, which is understandable because people have, you know, it's been awful. That virus jumping occurs on a regular basis, right, in the natural world, not just involving humans, involving every species you can think of. And humans give their virus to other things too. So humans are not we tend to think in disease emergence as humans as being the end of the chain, right? We're the last species. That's not. Humans can give their things to. I mean, humans. I mean, there's been big measles outbreaks, for example, in Great Apes in Africa, and that's human measles. Okay, so we've given those animals, they're dying because of it. So what I want to understand, I guess the goal of my research is what drives the diversity of viruses in nature, and part of one of the drivers is that they jump between species. So for a virus, all the virus wants to do, it wants to find a host cell to replicate in. Okay, so that's its home, that's its environment, that's got all its resources. Is this host cell? It doesn't actually matter in many ways what animal or plant that host cell is in, as long as it can find a cell to get into. So jumping between cells, aka jumping between viruses, being hosts, is the way how viruses live. And so what I want to try and do is understand the barriers and determinants of that process. And it's a process that occurs in all viruses and all hosts. So I look at these other animal species, in the chickens and the dolphins and the platypuses and the whatever, possums, to learn from them the general rules about how viruses do this, how are they able to jump between species boundaries. And and that happens all the time. Many cases the virus doesn't get going. Occasionally it can cause a little a transmission chain, and very, very rarely it becomes like a pandemic thing. And in nature, one thing I've learned actually in most animal species in doing this is that most viruses probably don't even cause disease. Okay, I mean, it's actually, so I sample a lot of birds, a lot of fish. I'm obsessed by sampling fish for some reason, right? So we sample a lot of birds and fish, and these animals are absolutely full of virus, right? They're full of virus. And so the fish sampling, for example, a lot of it at the local fish market. So you actually go and bought these. You can do this work in your kitchen. So you go and buy snapper, you go and buy whiting. You know, I bought loads. I bought a lot of fish. The great thing is you can barbecue your food afterwards. You have your research animal afterwards, and you sample these fish and you sequence them. And they're full of virus, right? Absolutely full of virus. And that's nothing wrong with that. There's nothing to be afraid of, because fish viruses don't replicate in human cells normally, right? But it just shows you that in the natural world, these healthy animals carry an abundance of viruses, okay, that are seemingly doing them no harm whatsoever. So the pandemic killer that we've just had, or still going through actually, is actually a rare thing, it turns out. So that my perspective, it's kind of changed by looking at the natural. We don't see it happen so often. So again, I want to understand the drivers of how viruses jump species boundaries, and that then helps us understand how viruses like SARS-CoV-2 can do what they do, or why other viruses. So I've done a lot of work in China in the past,
Yep.
and China has a lot of things called hantaviruses, very nasty
Yep.
viruses. And you get them in the US actually. So in the Western US, there's been lots of hantavirus outbreaks because these deer mice carry them. Now, the interesting thing about hantavirus is they jump between rodents and humans all the time. There's lots of jumping going on. So people in very, very famous outbreaks in New Mexico, Four Corners region get them, but they never jump. They never go from human to human. There's a one animal to human transmission, but never a big outbreak, never an epidemic. Why? Why are they unable to get going and become pandemic? And why is it that coronaviruses, this is like the 9th human coronavirus we found. How is it that they're so able to not only jump, but then to just keep going? What allows that virus to become so epidemic prone, in contrast to a hantavirus that's really nasty, gets into humans but never gets going? So those are kind of questions that I try and address. And to do that, because I'm an evolutionary biologist, I take a kind of comparative approach and look at all animals and all viruses, okay, because that gives us lessons.
And you brought up bats before, and somebody that you've published several papers with. I had on a show about 2 years ago now was Dr. Linfo Wang, and he introduced us to this Hendra actually down, which the situation was down your way, in that case bat to horse to human, one of the really interesting things as we see some of these, and actually when you meant the sort of the spill over and the spill back, when Dr. Zukasoko was on from Uganda, she was at that point in time, the first hippopotamus had just contracted COVID, so it was a kind of a sad day. But
is that right?
Yeah,
Wow.
it was a bizarre situation.
I hadn't heard hippo. That's extraordinary.
Yeah.
That's extraordinary.
But along those lines, whether we're talking Hendra or COVID, you published this paper, this was 2019 in November, called in Trends in Microbiology, focused on ecosystem perspective on virus evolution and emergence. And you sort of, in this paper, you add sort of what we think of the One Health concept, so, human to animal to intermediary, what have you, and you bring the ecosystem into play. And I think that's something we don't hear as much about, that whether we're destroying this forest or we're killing off this megafauna over here, it impacts everything else that allows a lot of this to happen. Talk a little bit about that paper, if you would.
Yeah. So, no, yeah, and thanks for bringing that up. And so I think virology as a subject has, I use the word suffered a bit from a very anthropocentric kind of view. Okay. And so for most of virology, we've been interested in the viruses that understandably infect us or infect the animals that we use as food, or the crops that we use as food, so or use in our daily lives. So the first virus ever found was tobacco mosaic virus, because it was a blight on a very economically important product. Of course, that wouldn't happen today, okay, because tobacco is no longer well, hopefully not a big thing for us. So there's been this very strong anthropocentric bias, and that's automatically kind of put humans at the center of the universe. It's kind of like looking at the history of science before Copernicus. So humans are the kind of center of the world rather than the sun. And the more I study the viruses in nature, that became obvious that this wasn't the case, right? That viruses are everywhere, and humans are just another species, right? A very disruptive species, a species that's kind of fueled more viral evolution than any other, but just another species. And like I said earlier, humans give their viruses to other things. So we're not the end of the chain, right? There's not this great chain of being that ends in humans. Humans are kind of linking it, and we can move viruses around. We can kind of connect species. So that leads to think, okay, rather than think about humans in this kind of linear way, right? Like the way you described it earlier, so you have a reservoir host, an intermediate host, and human, right? That's kind of got a directionality that humans are the chosen one at the end. It's not like that. And you mentioned SARS-CoV-2 hippos. It's a really good example. So humans have now passed their virus to an extraordinary number of species. It's just absolutely, in fact, all my 30 plus years of doing this, I've never seen a virus ever as host generous as SARS-CoV-2, as an aside, right? I mean, in whitetail deer in the US, amazing prevalence, right?
Yep.
So humans are, again, just one species in this kind of interconnected network. And I think what I think about disease emergence, so things jumping to cause, outbreaks, is they're often caused by a disturbance, the kind of a disruption to the ecosystem. And humans are the great disruptor, because what we're doing in the last, particularly post World War II, you've got massive deforestation, mega cities, global travel, these wildlife markets, all that just, species driving to extinction, climate change, human-driven climate change, all those things are kind of disrupting the natural world, putting animals into contact with each other that wouldn't have been in the past, putting humans into contact with animals that wouldn't have happened in the past. And the more you bring things together, the more you put animals together, humans with animals, you increase exposure, you increase the chances of these things jumping, right? So, the Anthropocene, what we are in now, humans have altered the planet. We're also altering the disease aspects of the planet as well. So I think we live in this completely massively connected world. And not just the way humans live, but the viral world is completely connected. So going back, I mentioned I was in this island in New Zealand. The idea there was to say, let's look at one ecosystem and let's see how often viruses jump between an interconnected ecosystem, right? So you take a snapshot of one population, how often are viruses moving? Rather than, what we normally do is we sample some bat viruses and some rodent viruses. We do an evolutionary tree. We try and work out, that one gave one to that one, that one. That's fine, but you're looking at a very long evolutionary perspective of thousands of years. What I want to know is, at one point in time, one interact community, how are they sharing viruses? And humans, there's no humans on Anchor Island in New Zealand, so actually humans are not part of this, but in other ecosystems, that virus sharing is going on between animals, and humans are pushing it, they're driving it. And as climate change goes on, we're going to see, so what's going to happen with climate change is that pristine habitats will decline, animals will be forced to group together. That will increase exposure. That'll increase viral jumping. Humans who rely on subsistence will need to go further into the remaining pristine areas to get their subsistence. They're interacting with more species. So, a very simple prediction of climate change is going to cause more pandemics. And I hate to break it to people, right? But this is going to happen again, and it's going to get worse. Okay, because we live in this ecosystem, because we perturbed it, that's going to cause more pandemics. Simple as that.
And thinking of the impact on, not directly on humans, but on things that supply us in the sense of food. This 2007 paper in AgriScience, where you studied something called Israeli acute paralysis virus, and this was right in the middle of when all the honeybee colonies were collapsing. Talk a little about that, because I'm going to get on the path here of sort of things that, this is not a human thing, but we need the bees, we need the honey, we need them to pollinate everything so we can eat food.
We need the bees. More than anything.
We need them. Talk about sort of analyzing some of these, dangerous things that are happening in the virosphere in terms of that, but at the same time, what are some things that, what happened as a result of that? I mean, are there solutions like, what the Linfa was telling us about in terms of Hendra? Okay, we vaccinate the horses, and that prevented the Hendra later on. But what can we do for something like the honeybee colonies?
It's a very good question. I'll just go back to Hendra. Actually, you mentioned a very interesting question, and this really shows you where it's critical to think about ecology. So, Linfa will say, rightly, vaccinate the horses. That's a really good. Obviously, it's a great thing to do. The other thing you can do, the ecological solution, is actually probably more important. Is Hendra comes from bats, right? And the problem is that humans have encroached on bat habitats. This is what's happened. It's the same as Nipah. You may have heard of Nipah in Southeast Asia, Bangladesh. So these bats are coming into these human villages, and they eat this date palm, and people drink this date palm. They urinate in this, defecate in this date palm sap, and people drink this, and that's what causes Nipah. But it's a very nasty disease. Now, what's the solution to that? Actually, it's great. What you do is you plant more trees. you plant trees away from where people live. You plant trees that bats want to live in. You make good habitats for bats where they want to be, away from human settings. You give them an ecological zone where they want to be. So they don't have to go into the date palm trees in your local village. Okay, they don't have to go in places where your horses are in Queensland and Australia. So if we improve the environment and give them a place to live away from us, you put a barrier to transmission. I mean, it's planting trees. It's as simple as that, right? Extraordinary, and that's an ecological kind of solution to it, right? Amazing, extraordinary thing. So if we think about that kind of way of thinking, let's these animal viruses that can infect us, let's give animals, get them away from us by making their lives better somewhere else. Okay, so it's a different way of thinking rather than, I like the high-tech solutions. Vaccines have been amazing in COVID. mRNA vaccines are unbelievable, right? We need those high-tech solutions, but low-tech ecological solutions, because the virus is part of ecosystems. If we manage our ecosystems there, we're going to do this, but we're also going to prevent pandemics. And ultimately, that's a more effective and cheaper way to do it. Okay. So in terms of like the bee virus, the bee thing, those of your listeners may not remember this, is that there's a thing called colony collapse disorder, which is a big thing. I lived in Pennsylvania at the time. And it was a huge thing. You remember these bee colonies were just collapsing and a whole going extinct, and that was that if we lose the pollinators, and of course bats, by the way, bats are important pollinators. We have to conserve bats; they're important pollinators, right? But if we lose the pollinators, we are in serious trouble. So it's critical that we have a healthy honeybee population. And so there's these colonies collapse disorders going on in the US particularly. And the question is, what was causing it? And so what that led to, which is actually very interesting, I think, is some of the first really proper virome studies where you go and you determine all the viruses in a species, okay? And that, I think, was one of the first kind of work on doing that. And since that's been lots of these virome studies. And so honeybees now actually one of the most studied of all organisms. We know the bee viruses quite well. In that case, back in then, it was still actually a little bit unclear what was really going on. We know there are these things called varroa mites. You may have heard of varroa mites. These nasty little mites that infect bees. They're all over the place. It's probably that the varroa mites were allowing viruses to come in greater numbers, and that was kind of stressing the population. There's a whole bunch of viruses that so it was the mite plus a virus that was doing it. Quite which virus is still a bit debated. So I think so. So the solution to that. So now we know that. So I think what the outcome of that has been. You asked about what the direct outcome of that has been. I think what we know that these mites are a real problem, particularly if there are viruses around. So we recently had an outbreak in Australia of mites. And so what they did, they set up basically a quarantine, a very strong quarantine around that area to stop the mites spreading, which meant it couldn't transmit the viruses, and the colony couldn't collapse. So that led to so that's essentially a kind of like physical distancing kind of solution, a kind of quarantine solution that we use for COVID. But at least we knew that what the genomics told us and the virome work is it defined what the problem was in more detail. And I think that was the first kind of case of ever that being done. As I said, we now do that. That virome work is now being done on lots and lots of species.
And continuing that thought with a somewhat related theme, you've published a few papers that were quite fascinating on sort of principles of viral biocontrol. One had to do with some nasty toad that was, I forget what the toad was doing, but it was bothering something. Yeah, the cane toads, this Rhinella marina endogenous retrovirus that were keeping that in order. And then there were some rabbits that were, I don't know if they were eating something that they didn't want them to eat, but here again, okay, so viruses have this beneficial effect. Okay, we don't want them to deal with get rid of the honeybees, but we might not want all these toads around and rabbits doing these nasty things. Talk a little about the principle of viral control. And we see that.
This is like the complete opposite, and it's quite a controversial topic. So, but it's one that is very interesting, and particularly if you live in Australia, because Australia has like the worst invasive species extinction problem ever. I mean, we're like one great laboratory for, human error, effectively. So I'll just give you the two examples that you mentioned. So one is the rabbits. So rabbits are not native to Australia, and they were brought in by the British, God bless them, I am one of those people, in 1859 into near Melbourne. And I think there were 24 rabbits that were brought in. By about 1950, so 100 years later, it's hard to know the number, but it may have been close to 1 billion rabbits, right? So they literally bred like rabbits. And it was the fastest growth of any vertebrate and spread of any vertebrate species in history. So they're pristine environment. There are burrows from native marsupials they could use. They just went crazy. So and they call it the Great Rabbit Plague. And there are literally these waves of rabbits. And you may have seen a movie called Rabbit Proof Fence. If you haven't seen the movie, by the way, fantastic, horrifying movie, fantastic movie. So they built these. So one thing they tried to do is build these fences across the country to stop the rabbits moving. And there's one that goes from kind of Western Australia to Northern Australia, which is the movie is set on. And they're literally, I've got a photograph somewhere of the rabbits who got to the fence, and they're like 25 foot deep of rabbits because they're backed up at the fence. They can't go any further, just a sea of rabbits. And these sort of, the birds come in, birds of prey come in, just picking the rabbit they want because there's so many to choose from. It was a massive ecological problem, and it's destroying the grasslands. And native species were being outcompeted, that were burrowing. We've lost a lot of mammals in Australia, native mammals. So they spent a lot of time trying to work out what the best solution to this problem was. In fact, at the end of the 19th century, there was a prize because they were so worried even then, they announced an international prize, I think it's like $10,000 to find the solution to the rabbit problem. And Louis Pasteur entered. Louis Pasteur entered his cousin. He sent his cousin over, and the idea was to introduce foul plague, Pasteurella bacteria, to try and kill the rabbits. And they built a little research station in Sydney Harbour. They bought an island, Sydney Harbour, built a research station. This is actually the first ever Institute Pasteur. Is in Sydney Harbour, Australia. Little history, amazing to try and. He didn't win the prize. Anyway, so he went on. And then in 1950, they were so desperate, they decided to release a virus called Mixomovirus that causes myxomatosis. And this virus is native to a South American species called a tapete. It's kind of rabbit-like thing. And it kills them. As in the tapete, it actually causes these lesions that are quite benign, actually. But in the European rabbit, the one that is in Australia, these lesions are lethal, and it's passed on by basically mosquitoes, by vectors. So they released this virus in 1950, and the mortality rate initially was 99.8%. Rabbits died. Can you imagine? It's just an extraordinary. I don't think it's almost the most lethal virus ever. I mean, incredible. And it just killed a sea of viruses. He literally had fields of just dead rabbits, dead rabbits everywhere. This virus, so devastating. And it had no kind of detrimental effects. The researchers that worked on it actually injected themselves with it, amazingly, to see it was safe on humans. But then what happened, this is why I got involved, is the virus then evolved. So initially it was massively virulent, but that virulence kind of went down, so it became less. It's still actually pretty nasty, but it became less nasty. And the rabbits evolved resistance, as you'd expect. So there's a massive selection on the rabbit to evade this virus. So it's the textbook story, of host virus coevolution, how virulence evolves. So it worked. It dampened the population pretty well, but didn't go extinct. They then released the second virus in 1996 called rabbit hemorrhagic disease virus, a Calisi virus, that also killed an enormous. These are not engineered viruses; these are natural viruses that caused an enormous mortality event in rabbits. Again, no detrimental effects, reduced rabbit population, but the rabbits are very robust. They're still kind of there, but these biocontrols have actually been pretty successful. Now. The thing about Australia is that we don't have really almost no native placental mammals. We have marsupials, but not placental mammals. And so the release of these viruses is a little bit more little bit safer
Sure.
than you might expect in other locations. It's not going to jump into lots of it. Hasn't. We don't think it's jumped into any species so far. The problem, of course, with biocontrol is it does work. It's a very, you know, a very dramatic solution. But the worry is, would it jump into other species and cause other problems? This is the great concern. The one they're concerned about now is actually carp. So Australian rivers are full of European carp that were imported in for fishing. So there's a thing called the Murray-Darling River Basin. So Southeast Australia's got one massive river system, and now I think like 90% of the biomass of fish is one species, European carp. And so there's been lots of solution about how do we. It's like the rabbits, right? But
Sure.
fish. How do we remove this carp? And one of the ideas was to release a virus, a herpes virus that's found in ornamental fish, koi. Yeah. So koi herpes virus, and to try and release this. They called it Carpageddon, to try and destroy this amazing carp. Now that hasn't happened. The proposals were made pre-COVID. There's lots of safety testing being done. I don't think it will actually happen. I think the worry would be, post-COVID, I think we're in a slightly different thinking about viral diseases. I think there'll be a lot of dead fish around, and that will itself impact the river system. So I don't think that's going to happen. So biocontrols are they are a solution, but they're not one to be used lightly. And but yeah, they're fascinating. I'll just say one more story very briefly. The first biocontrol ever used, actually a fascinating story, is in a place called Marion Island. Have you heard of Marion Island? So Marion Island is in the South Atlantic. It's just north of Antarctica. It's between South Africa and Antarctica. Okay. It's a vast stretch of ocean there. These are French-owned islands. So really, really remote, windswept, bleak kind of places. But there's a weather station on Marion Island, and they had a rat problem because of the weather station, right? So rats are there. People rats. There's rats everywhere. So they introduced some cats. You know what's going to come on later. They introduced some cats to kill the rats, right? And by about 10 years later, there's like hundreds of cats, literally or thousands of cats probably. And the cats are then eating these rare seabirds that come onto the island. Again, it's humans playing with ecology, right? That's all it is, So there's these rats and there's the cats and they're killing the birds. So they introduced a virus called feline parvovirus to kill the cats. And it worked. And they massively reduced the number, and they trapped the rest, and they removed the cats from the island. That was the first ever use of biocontrol. Now, that was actually, you would never ever want to use that virus because it jumps between we now know it's the most jumpy virus you can think of. It goes between loads of different host species. If you released it in North America, you would lose lots of native carnivores. So it's a disaster. Luckily, this is the most remote island you can think of in the South Atlantic, right? So it was okay to do it. But it just shows you these ecological effects that humans have. Absolutely. And how viruses kind of play a role in that.
Absolutely. That's a fascinating story. I was unaware that even existed, but no, that's a good one. Hey, one of the looking back sort of at some of the early days in your publication history, back in actually February of 2001, you published a paper on hepatitis G. Virus, and this is interesting because both I had Jack Stapleton a few years ago on talking about it, and then from your old neck of the woods of Penn State, Maryland, Rusink, and here we're dealing with you brought up the virome before, but we have these important stories. I mean, this is one of the ones that we've heard a lot about. Here's something that has a beneficial effect in the sense that, people infected by this have a lower risk of whether it's HIV or herpes infection and so forth. We've also done shows on bacteriophage viruses and so forth. Talk a little bit about some of what you find when you're sort of, once again, swimming through the virus sphere, whether it's things like hepatitis G or other interesting beneficial viruses, because once again, there's a huge part of the virus sphere that is not dangerous to us.
Yeah, I think beneficial is it's a harder thing to actually quantify. I
Sure.
certainly think that most of the Virosphere is neutral. It's not doing anything, right? Certainly in animals too. Humans are actually very interesting. We don't have that many viruses that are non disease causing. So why humans are different is an unusual question. So hepatitis C, that's now called GBVC,
Right.
by the way. So it's got a new virus. That is a commensal, a benign virus. And humans have 3 or 4 of those. If you sample people, 10% of people have GBVC. Or it's actually now called human PEGI. They've changed it again. It's now called human PEGI virus. Right? It's got an even newer name.
That's cool.
So you find that virus all over the place. The evidence that it's beneficial, I think, is pretty scant. I think it's certainly not doing anything dangerous. But there are very few of humans have very few of these non-disease causing viruses. Most of the viruses we have causes. Now, whether it's because of the way we vaccinate, whether it's because of our hygiene, we're not exposed so much, but humans are kind of unusual. In the natural world, animals that I work on, you see lots of viruses. We see no evidence of them actually having any detrimental effect at all. How many of those are beneficial? That's a very hard question to answer, because you need to look at very complicated interactions between them, and people don't do that kind of research. The benefits might be, for example, if you have immunity to one virus, that may help prevent something else coming in that's actually quite similar. Okay, so we know that dengue and yellow fever, if you have antibody to dengue, that might help prevent yellow fever kind of thing. So that kind of that might be a reason. There might be some proteins that are expressed that help prevent other viruses. I think it's a very good question. I think the way that the field is going, I think we've moved away from the idea that all viruses are necessarily bad and causing disease. I think we moved away from that. In the future, I think one of the great research themes will likely be how many of them are beneficial. Okay, I think that's kind of where we're going to go. I don't think we're quite there yet, but that might come up.
Yeah, and I think with everything we hear about what's going on with the microbiome and sort of microbiome living therapeutics and so forth, I think it's going to be a piece of that.
Yeah, and so phage therapy, bacteriophage therapy, we use bacteriophage to kill bacteria. Yeah, that's a really exciting area. I completely agree. That's a very interesting thing, and we don't know. And so when I say humans, I should say, qualify a point. When I mentioned human viruses, I'm not talking about bacteriophage. I'm talking about viruses that infect humans. Of course, bacteriophage infect bacteria within humans. So I don't class as human viruses.
Gotcha. Eddie, can you say a few words about? I know you had. I've read some articles. You've been quoted in when referring to sort of the predict spillover tool. John Mazzette was here about a year ago talking about that. I know you had some opinion in terms. It's great to sample everything, but there's a lot out there, and maybe more of a targeted surveillance versus just cataloging everything. Talk a little bit your perspective on this, please.
And so our difference of opinion, and it's not a big thing, I don't think. I mean, certainly we're mainly on the same page, is the fact that I have no problem. I work on virus evolution diversity, so I do a lot of animal sampling. I mean, absolutely. I think that's very important to understand how viruses jump species boundaries and how they evolve. What I don't think that tells me a lot. It tells me where viruses come from. There are the rules of how they evolve, the rules that shape the diversity, how often they jump species boundaries, the barriers to that process. It tells me that. It doesn't tell me, I don't think, what's going to jump next. Okay, so my problem with biodiversity studies is, I think they're fine as they are. Do they give you predictability? They give you something tangible. You can say, aha, that's the next pandemic. And I don't think they do. And the problem, and I don't think they do for a number of reasons. And the main one being reason, there are a huge number of viruses. And if you look at a bat species, for example, everyone's obsessed by bats. If you look at bats, it depends. There are an awful lot of bats. It depends which bat population you look at, which part of their home range you look at, which season you look at, because viruses have a seasonality. So if you look in the spring, you look in the winter, you might see a different set of viruses. What tissue you look at, okay? So We often use fecal sampling for bats because it's safe, right? You basically just collect bat poo with a tarp right under a tree. It's kind of easy to do. But if you do that, you mainly get dietary viruses. You get the insects and the plants that they've eaten. If you look at bat tissue, if you look at a liver of a bat, something like that for a diseased bat, you get a different set of viruses and you look to do fecal sampling. So it depends on place, time, what tissue, and they evolve rapidly. So if I did my sampling of bats today, I looked in 5 years' time, there'd be the virus has evolved on. So there's an enormous number of viruses. So to me, to say which of that extraordinary large virosphere are you ever going to predict that's going to cause disease? Also, they're going to evolve in the host. Okay, so SARS-CoV-2, we have bat viruses that are quite close, but there's still an evolutionary gap, and there are bits of sequence in the human we haven't quite found the ancestry of. Right. So if you found a SARS-CoV-2 like virus in humans back in 2019, what would you have done? Would you have said, "Oh no, I'm going to kill all the bats. Am I going to vaccinate them? Am I going to evacuate the people from the local area?" Or would you have done nothing? I bet they would have done nothing, because they wouldn't have realized that that virus had characteristics that were going to do anything. So I don't think that mass sampling of animals in nature is necessarily predictive. What I would do, as you mentioned, is more target surveillance. I think the human-animal interface is the place. I think it's people who work at, in the wildlife trade, in bushmeat, in abattoirs, in animal markets, veterinarians, animal carers who interact with animals. They are on the front line. They're the canaries in the coal mine. They're the people being exposed, right? So those people and the animals that they're interacting with, who work in, and I've done a lot of animal market sampling, right? Wildlife trade sampling. They're the risk. They're the fault line. They're the stress point. They're the risky point, right? So I focus on them and then have really good surveillance. Now that necessarily means we miss the first case, right? I think you're gonna get the third person. I don't think we can ever prevent that though. So I think if we have really good target surveillance, I think we've got a chance of preventing the next pandemic. So if you go back to COVID, right? I don't think we'd ever prevented that spillover happening in Wuhan, but I think we could have prevented the pandemic. I think if we'd acted quicker, we could have stopped the going pandemic. Okay, so I think there are. Once you've got early information, I think there is time to act. I think we dropped the ball there, and we've dropped the ball so many times in the past.
Eddie, in 2010, I think that's when it was formally established that it was Yersinia pestis that was the actual cause of the medieval Black Death. You published a paper in 2020 in Current Biology on the importance of studying these ancient pathogen genomes and some of the things that we can learn. Talk about sort of, where this potentially goes. I mean, is it studying mummies, or I mean, what potentially is the scale by which we can search in the sense of this virosphere again, going back in sort of the human experience?
Yeah. This is a kind of side project. Look, and before I go on, a lot of people get overly worried about this sort of work. This, as it says, things are very completely fragmented, right? They're degraded to anything. These ancient samples, they're at no great risk, right? So, a virus you get from a bone from Black Death in 1347 is not going to give you pestis, because it's completely degraded, right? So put that out there. I think what it tells us is, so for that particular, this is bacterium Yersinia pestis, not a virus. What we're interested in was trying to work out the virulence. Why is it that these older plagues, so the plague, the Black Death, a pandemic of Justinian, the Roman emperor in 540, killed half Europe's population. Why were they so nasty compared to the modern plague outbreaks? They're a lot more benign. What is it? Was it the bacteria? Had the bacteria changed? Were the bacteria was the Black Death just an intrinsically nastier thing, or was it, this is actually I think the right answer, that people just lived in squalor, right, effectively, and then they were continually exposed, they were unhealthy, lived in overcrowded cities, had awful hygiene, awful diets, and so the bacterium was enough to take them over the edge into the disease, rather than being an intrinsically virulent thing in it. So I think it's more likely as a combination of factors that and the bacterium added something than it being a highly virulent strain in itself. So I think what this tells us, in that case, it can tell us about virulence. The same's been done for like smallpox. Okay, so with smallpox, we think of as being, we know for smallpox from Epidemiology, of course, and cowpox vaccination. So our consciousness of smallpox starts in 1796 with Jenner, but now they've gone back and found smallpox back in the Viking times. Okay, it's incredible, right? So way older. And again, the question is, can we see signatures in the genome that tell us how virulence might have evolved? What are these? Was it less virulent, more virulent? Has virulence changed through time? So I think that's the most interesting thing, is actually that because you get so few of these ancient samples, you can't really construct. I don't think you can really construct complicated popular histories, but I think you can sort of say, can you see gene changes that may be responsible for evolution of virulence? So I think to me that's the really thing that this tells us. The gap we've got, I'll tell you, the gap we've got with ancient pathogens is all the things we mentioned so far, all the things have been done. Smallpox, plague's been done, hepatitis B virus has been pushed right back, leprosy, mycobacterium has been pushed back. They're all DNA-based organisms. What we haven't got are ancient RNA pathogens.
Got it.
The oldest thing that I really think people believe in is called 1918 flu, because RNA degrades really quickly. So the Holy Grail out there, listeners, viewers, is to get a way of looking at ancient RNA. That would be really interesting. And again, it's the start of around the early 20th century is kind of where that field. I'd love to get ancient RNA viruses out there to see what they look like, but they degrade. They degrade so quickly, it's hard to do. But that's the gap, and technology will get there, I reckon. But we'll see.
Yeah. It did in Jurassic Park, but they never.
Yeah, it's interesting.
They never explained that part about the RNA, but
yeah.
Eddie, I have to ask, because again, I went through, I spent time sort of swimming through your publication, and this one popped out at me, and I would love to hear, as we get close. I know you have a hard stop in about 13 minutes, but I would love to hear the story about the phone call that you got probably maybe late 2018, early 2019. Somebody calls you up and says, Eddie, I have a thoroughbred stallion that has a lesion on his penis. I have semen. I want you to take a look at it. What was that all about that led to the publication of this August 2019 paper in Viruses?
Oh, wow, that's the first time anyone's ever asked about that one. Yeah, so one of my good colleagues in Australia is a GP, a doctor I work a lot with, called Bernie Hudson. He's a great disease physician in northern Sydney, and he's got a sideline of breeding horses, right? Race horses, not very good race horses, I have to say, but race horses nonetheless. He doesn't make much money, but and he works a lot with the kind of stud industry in Australia. And this is, I didn't know anything about this, but this is a hugely big money industry, right? So these mares they're serviced by a stallion on a regular basis, and you pay a lot of money for a service. Okay, it's like, I don't know, it can be. I think the most one I saw was something like $250,000 per service, right, for the top bred. I mean, it's crazy amounts of money, right? So so there's a huge amounts of money in this. So if the stallions can't perform, they can't do their servicing work, it's a big thing, right? Yeah. So there was a mid-ranking horse, the name of it slipped me from what I probably should name it anyway, that was having problems in the servicing area, and we wanted to take a look at it, what it was, and he had this lesion on its penis, right, its main servicing tool, and so they wanted to know what the cause of that was. So we sequenced it, and there was a papillomavirus, which is, so people are used to papillomaviruses. They cause genital warts, and actually very serious. So cervical cancer is caused by a papillomavirus, right? And there it was. This particular, we think that was the cause of this particular lesion, preventing the stallion from doing its job, right? But it was a big money. Unfortunately, one of the great things, I wanted to name the virus after a certain individual who was a well-known recent British Prime Minister, but I was not allowed to under the rules of taxonomy. So unfortunately, there you go.
A day in the life of the evolutionary virologist. No.
Oh, dear, it's excitement every day, all right,
All
every
right.
day.
Eddie, what else is hot? What's coming up for 2023? Anything hot that I missed? Conferences you're going to be presenting at? Talks you're going to be giving?
Well, so
Please take the floor.
yeah, the main thing that we're going to do, I think, is more of this. We touched on the COVID issue, and so I think, briefly, I think what I'm particularly interested in now, I've mentioned it a few times, is the wildlife trade and the animal farming and what that leads to. And so, with colleagues in China, we are still looking at outbreaks in these animals that are farmed and then go into these animal markets. Now, animal markets are still closed,
Mm-hmm.
but the breeding places are still there. So at the moment, we have a variety of outbreaks in things like raccoon dogs and mink in China that we're working on to work out what the cause. I mean, these animals are kept under not very good conditions, and there's lots of disease outbreak. And to me, that's the front line of emergence. So we're trying to work out what are in those things. We've got a lot of sampling going on. I think these are pretty big studies. I don't quite know what's in them yet, so I can't give you any kind of tips. But we're trying to kind of work. I think that to me is a crucial thing to try and work out. It's where humans and animals come together, and that's where you're going to get risk. So I'm going to do more of that. I'm doing more of that in the Australian context too. I've talked about ecosystems in New Zealand, but also do my local kind of ecosystem, because in Australia, humans are going more and more in building into the kind of the wild area, interacting species that carry diseases. So I'm interested in that nexus between humans and what I call backyard wildlife that we interact with all the time to see what's jumping. So it's the human-animal interface that I'm working on, really, and hopefully there'll be some big stories. Hopefully not too big, no big outbreaks. I'm hoping it's no big outbreaks, but
no more penis
hopefully we'll see. We'll see what's being, see what we're being exposed to on a regular basis.
Excellent, really excellent. Well, I wish you a successful 2023 as we get close to the new year here. For everybody that is going to be listening to this particular episode of the show across the various podcast networks or watching on the YouTube channel. Again, you've been listening to Dr. Eddie Holmes, Professor, Sydney Institute for Infectious Diseases, School of Life and Environmental Sciences, and Sydney Medical School down at the University of Sydney in Australia. Eddie, I want to thank you for taking the time out of your schedule to come talk to us for a little while about everything you're doing there. Obviously, thank you for doing it. And as we say on our show here, thanks for helping to create a better tomorrow through the research you do. Really fascinating stories.
Thank you so much. That was a great pleasure.
Good seeing you.